Okay, so today we're going to discuss antibiotics and then some decontamination methods to kill microbes. All right, to start off, antibiotics. These are a classification of drugs that help mitigate bacterial infections. So there's generally two types of antibiotics, and the first one's bactericidal, which means, side, that ending means to kill. So these guys kill bacteria, and then bacteriostatic. Static means to be still. So they stop bacteria from growing further, but they don't necessarily kill the bacteria that are already there. So there's a lot of different types of antibiotics that'll be used commonly, and they'll all target different parts of the bacterial life cycle. And actually, a lot of them come from either bacteria themselves, so bacteria wanting to kill other bacteria near them that might be competitors, or by fungi or just other organisms that might want to inhibit the growth of bacteria around them. Yeah, so the first one, these are pretty much the most well-known type of antibiotic. They're called beta-lactam antibiotics. And what they do is they bind, let me get my pen here. They bind to penicillin-binding protein / transpeptidases. And these are the enzymes that help synthesize the cell wall. So they create that peptidoglycan structure inside the cell wall. And what they do is they mimic what's called the D-alanine portion of these precursors to the cell wall that needs to be added to help make the structure more rigid in the cell wall. And I'll draw that out on the next slide, so it'll be more clear in a second. But basically, they're messing with the creation of the cell wall, maintenance of the cell wall of the bacteria, which means that the bacteria will die because the cell wall will break. And some types include penicillin derivatives. I'm sure you've heard of penicillin. The first antibiotic that was discovered, actually. And we have cephalosporins, carbapenems, and monobactams. These are just names of different types of beta-lactam antibiotics. So let me draw out real quick what's going on here. So if you remember, just to refresh, all right, peptidoglycan, remember, it makes up the cell wall. And if you remember, it's made up of these sugar monomers, all linked together in these long chains adjacent to each other. And if you don't remember the names, these are, remember, NAM, can't really see here, NAM, NAG alternating, remember, N-acetylmuramic acid, N-acetylglucosamine. These guys are alternating sugars that form these long chains. And essentially what they have is they have these amino acid components sticking off of them like this. So it's actually 4 here. So they have 4 amino acids sticking off of each other. And then between chains, there's another 5, 1, 2, 3, 4, 5. That should be behind that sugar there. But there's another 5 amino acids that essentially link together these chains here like this. So when you're assembling the cell wall, let's say you're trying to build it up here. What you do is you have a sugar with 5 amino acids coming off of it like this. And then as you add it into the structure, you basically cut one of them off, and then you're left with these 4 here. And then 5 other amino acids will come in and link the 2 side chains together like this. So in most bacteria, these last 2 amino acids are D-alanine and D-alanine. So basically, we need these D-alanine regions to come onto this guy here for the new part of the cell wall. I might have some membrane before, that's my bad. It's the cell wall to assemble here. So what these, and then the enzyme, right, the PBP, the penicillin binding protein or / transpeptidase, this is the one that puts these amino acids into the structure so that this can go and make the cell wall. But what these beta-lactam antibiotics do is they mimic the shape of these D-ala portions here. So the penicillin binding protein is now, it's going to bind to I'll just write A for antibiotic. It's going to bind to this guy instead of binding to the D-ala to put it here. So this is basically inhibiting the action of the penicillin binding protein, which means that none of this stuff is going to happen and the cell wall can't be assembled. So hopefully that makes sense. Let me know if I need to explain that further. Yep, so of these, like I said, penicillin is the first antibiotic and the most famous, probably, that you've heard of. And it was discovered in 1928 by Alexander Fleming. So this guy, he's a scientist. He's doing his research. He has a bunch of plates of bacteria, right? So like petri dishes. So he happens to leave one in a cabinet on accident. And he leaves it there for, I think, a few weeks, I believe. Then he comes back, and then he sees that there's some mold that has grown on the plate, right? But then he also notices that where the mold has grown, so you have this plate here, you have the bacteria on there. You have this, let's say this mold has come and infected the plate. Now he sees this region, this ring around the mold where no bacteria can grow. So outside of here, the bacteria is growing. You can see that all that darkness, dark culture showing up. But then this one region is just blank. There's nothing growing there. So this is actually because this mold naturally produces penicillin as an antibiotic, because for some reason, the mold doesn't want the bacteria to be near it. So it basically produces penicillin and creates this, what's called a ring of inhibition, where no bacteria can grow near it. So this is where penicillin was first discovered. Then, of course, after it was discovered, they started using it. Then, during World War II, it was a huge help because the soldiers can now treat bacterial infections like on the field or like in the field with penicillin. So, and from there, we just kept going and going, and then we developed all these different derivatives of drugs that are related to penicillins. All right. So next we have tetracyclines. So these are these are bactericidal, just to clarify, the beta-lactams. Right, next we have tetracyclines. These are bacteriostatic, so they stop bacterial growth. They don't kill the bacteria. So what they do is they inhibit protein synthesis by binding to what's called the 30S unit of the bacterial ribosome. So if you remember, the ribosome kind of looks like this, right? And then when the mRNA is coming over here, right, after transcription, mRNA kind of inserts itself here, and then you get this ribosome moving. So the ribosome basically has two subunits. You have the 30S and then the 50S on top, like this. So I know it's weird because technically we know that bacterial ribosomes are 70S, right? Remember Svedberg's measure how well the molecule how well its sediment sinks to the bottom in a centrifuge. If you don't remember that, go back to the bacterial internal structures video, I believe. So, or the slideshow, sorry. But even though this is 50S and 30S, it's supposed to add up to 80, right? But it adds up to 70. This is just because the Svedberg unit isn't necessarily linear, because these things independently can sediment at different rates compared to the overall ribosome as a whole. So just know that 50, 30, but the whole thing's 70. And an example would be doxycycline, so anything that has cycline at the end of it. All right, macrolides, these are also bacteriostatic, so they stop growth. And again, they inhibit protein synthesis. This time they bind to the 50S unit here and inhibit its function here. An example would be erythromycin. And these are just, you don't have to know these names. Just, I thought it'd be cool if you've heard of these drugs before. You might, it's cool to know how they work. All right, glycopeptides. These are bactericidal, right? So they kill the bacteria. And so again, you're going to be messing with the cell wall here. And this time, so remember before, for the beta-lactams, we were binding, beta for beta-lactam. We were binding to those penicillin binding proteins that help assemble the cell wall. This time what we're doing is, instead of binding to the protein itself, we're binding to the substrate of the protein. So remember, we had the D-alanine here that this guy needs to get and then incorporate into the cell wall, right, onto these, and then et cetera, et cetera, et cetera. But instead, what they're doing is that they're binding to the D-alanine itself. So the glycopeptide, I'll say G, will come bind here. This will prevent it from being grabbed by the PBP and prevent it from being incorporated into the cell wall. And again, this causes cell wall breakdown, which the cell will just explode and then kind of dissolve away. And an example of this is vancomycin. And vancomycin actually pretty cool. It was isolated from a fungus in the sewers, I believe. So, some of these crazy compounds that are super useful for us can come from like the most unexpected places, which I think is pretty interesting. And it's like from another microbe as well, not like something a human made. So it's pretty cool. All right, fluoroquinolones, these are bactericidal, and they bind to topoisomerases. So if you remember from my last video, right, you had when DNA replication is occurring, right, et cetera. Remember, you have to stabilize because these guys are like spinning around. They want to condense again. So you have that topoisomerase enzyme sitting right here to stabilize this replication fork and make sure that it stays open, right? So when fluoroquinolones, they bind to the topoisomerase, they inhibit its function. Now the DNA replication can't happen properly, then the bacteria can't divide. And they eventually die because they, eventually they need to split the cell, but, they're just like sitting there until they die, basically. All right, so sulfonamides or sulfonamides, these are, bacteriostatic. So they're stopping the growth. And what they do is they block a specific enzyme. If you have this pathway, a series of proteins that create subsequent molecules, and then tetrahydrofolic acid. So there's this long pathway with a bunch of different enzymes that creates, eventually in each step, you're converting the previous molecule into a new molecule until you get this tetrahydrofolic acid. So a sulfonamide goes in and blocks one of these enzymes. And then this is the universal sign in biology for inhibiting, this arrow with the dash thing on the end like this. So these guys are inhibiting one of the enzymes so that tetrahydrofolic acid can't be made. And this tetrahydrofolic acid actually goes and becomes a nucleic acid. So essentially when you add this antibiotic, you're stopping nucleic acid from being made, so you can't make DNA, RNA, that stuff, which means that the cell can't develop, so it's just kind of sitting there on its own. Okay, so one big issue for antibiotics is that they target gram-positive bacteria pretty well, usually because, like we talked about, we had those beta-lactams and those was it glycopeptides? Correct. Yeah, the glycopeptides that bind, mess with the cell wall and make it explode. So you can always target the cell wall, and that's a pretty good bet because the gram-positive bacteria have that thick cell wall. So once it's broken, everything's just going to explode out. And then again, there's only one membrane, right? So the antibiotics can kind of penetrate easier into the cell. Whereas for gram-negative bacteria, right, you have two membranes. So remember, membrane 1, membrane 2, outer inner membranes. Then you have this thin cell wall in between. Oops, I don't know why that happened, but okay, we'll ignore that. Yeah, so they have two membranes, first of all, so it's harder to penetrate through. Then again, remember, we have these lipopolysaccharides, LPS, sticking out of the membrane like this. And remember, these can help with toxicity, so they can aggravate the immune system, and then they can also help with adherence to surfaces and etc. But this kind of creates this mesh framework that's very hard to penetrate through. So a lot of drugs will just get caught up in all these fibers here. So this means that gram-negative bacteria are harder to target with antibiotics. So there's a lot of different methods, but one that I'll just mention is this specific drug called colistin/polymyxin E. These are just two names for the same thing. So essentially, this is a bactericidal antibiotic that specifically can help kill the gram-negative bacteria, and that's because it causes the destruction of the outer membrane here. So the membrane itself is, as you can see, disrupting outer membrane, so this stuff is all broken, which means that this antibiotic can get in here a lot easier. And this is actually what's called a last resort drug because there's some potential for it to be toxic for the kidneys in the human body. So it's kind of, when everything else doesn't work, this is kind of the last thing you go to, and this will pretty much work every time for gram-negative infections. And then we also have aminoglycosides. These, again, similar mechanism to what we looked at before, which bind to the 30S ribosomal subunit, and these also kill the bacteria. And these guys are also pretty effective against gram-negatives. All right, let's talk about some limitations of antibiotics. So you might be thinking of antibiotic resistance, right, immediately, so we'll get to that in a second. The first thing is that antibiotics, they're not highly specific. So this means that when you take an antibiotic, right, you want to kill the bacteria or stop the bacteria from growing that are actually harming you. But you also have all the bacteria that are normally in your microbiome that are helping you, they can help you make important chemicals like vitamins or necessary fatty acids, or they can help you with digestion. There's a huge role of all the bacteria in your body. But the antibiotics kind of work so that a wide variety of bacteria will get killed in your body. So it's quote unquote the good bacteria can die too. So this means that we have what's called dysbiosis, which is when the microbiome, the bacteria, the microorganisms in our body, the balance gets disrupted with like how many of each like type there are. And that can cause a lot of different health problems. There's actually very interesting research. So if you just search up like microbiome or gut-brain axis, there's a lot of interesting things here because just to go off on a tangent a little bit, there's some evidence that like the bacteria in your gut and their function determine your like cognitive functions as well as some of your brain development. So there's a lot of like interconnected things that you might not expect to be connected. So if you're interested, you should search up gut-brain axis and read some papers, look at some articles. It's a pretty interesting topic here. So we don't want that to happen, right? So there's some research that has gone into what's called phage therapy. So this is when we use viruses, remember phages that only kill bacteria, and we kind of inject them into your body so that these phages are highly specific because they can be kind of engineered so that they only target a specific strain of bacteria in your body. So this means that phage therapy is very highly specific therapy that would probably get rid of a lot of the effects of using antibiotics and causing dysbiosis. So there's some research going on with this. They kind of used it in the Soviet Union like 60 years ago or something, but up till now, there's not been too much like uses of this like publicly. First of all, because I believe it's relatively expensive to do. And then second of all, there's a lot of stigma with, right? I mean, we know these phages only kill bacteria, but when you tell a patient that you're going to inject viruses into them, like I don't think anyone would willingly sign up for that. So that's kind of another issue that's at hand, but there's some research going into this as well. Yeah, and finally, the big one, right? Antibiotic resistance. So the overprescription of antibiotics. So basically, doctors they're prescribing antibiotics even when they're not necessarily needed. They're just kind of giving them out as like a general treatment to everybody, because if a patient doesn't return with a prescription, then they probably feel like they didn't get treated well enough, right? So that's like one of the problems here. But essentially, basically, it's like the overuse of antibiotics is driving bacterial evolution, because all the bacteria that are susceptible to antibiotics are going to die quickly since we're using them a lot, which means that the ones that survive are going to be the ones that become resistant due to mutations, right? Or horizontal gene transfer, as we discussed in the past. So yeah, essentially it's just like kind of arms race between antibiotics and bacteria. As antibiotics get stronger, bacteria will develop more resistance to them. Then we have to make stronger antibiotics, and then those will create even stronger bacteria. So it's just like this really bad back and forth dynamic that's going on. So this is a really big problem that so many researchers are working to combat right now. So yeah. All right, so we covered antibiotics. Now let's look at some decontamination methods. So ways that you would, disinfect surfaces, medical equipment, all that stuff. Yeah, so this term decontamination is a general umbrella term that kind of encompasses everything where you're trying to get rid of microbes from a surface or from like skin or something like that. First we have these levels. Sterilization is the most like intense level of decontamination. This is where you're trying to kill every single microorganism and virus. Well, you can't really kill viruses, but you're kind of getting rid of them on a surface or object. So you're trying to get rid of 100%. So you can see down the right here, sterilization has an empty visual here. Then disinfection is like one level down from that. So here we're killing almost all microorganisms, except for sometimes what we call endospores. And we'll go into this on the next few slides, so stay tuned for that. But essentially endospores, these are like resistant bacteria that are very hard to kill. So this infection is like a little bit less comprehensive. Then we have even less comprehensive is sanitization. So this is just like using hand sanitizer, right? It's killing like the vast majority of the microbes, but they're still leaving a lot of them there technically, but it's just at a quote unquote safe level that we've defined is like okay for us to have. So you might see like that kills 99.9% of germs, so there's still that 0.0 or 0.1% left over, right? And then we have antisepsis. This is just a term or a general term for killing microorganisms that are on skin or tissue. So we're actually cleaning like human, part of a human here, not just the surface. Whereas these previous ones are referring to cleaning inanimate objects or equipment and stuff like that. All right, so let's cover endospores. So endospores, these are these special structures that some bacteria can form, and they're extremely resistant and dormant forms of the bacterium. So the bacteria aren't dividing, they're kind of minimizing their functions. It's like they're hibernating. They form this thick shell around themselves so that they become really resistant to factors like heat, radiation, freezing, desiccation, which is like drying out, so lack of water, and then like chemicals. So these guys are really hard to kill and pose a very significant problem for sterilization. Yeah, and then some terminology. For the bacteria that can form endospores, the kind of normal form of the cell is called the vegetative cell here. So you can see that on the left, you see these vegetative cells, right? They're just normal bacilli. Remember the kind of oval-shaped bacteria. Then when they form these endospores, you can see there's kind of condensed down into that thick shell kind of shape here. So let's look at let's look at the structure of the endospore, then we'll look at how this process actually occurs. So endospores, like I said, they're surrounded by a thick coat of several layers of densely packed proteins. So you can see that here. We don't have to look at all the separate layers, but you just see these concentric circles of like protein here, essentially. And this coat is what gives the bacterium this like strong resistance to environmental stress. So within the coat is protecting what's called the core. This core is dehydrated, has not that much water, and it contains all the essentials that the cell needs just to maintain life, like at the bare minimum, pretty much. So it has all the DNA, like it has its genome, has some ribosomes so that can make some proteins that it needs. It has some other essential components as well, maybe some lipids, and then it obviously would have the building blocks like nucleic acids, amino acids, etc. And this is essentially suspended, and it's surrounded by what we call calcium dipicolinic acid, or DPA, CADPA, either one. So this acid actually, what it does, it gives the cell a lot of heat resistance, because what it does is, so this pretty much most of this core here is actually floating around in DPA, but DPA can also insert itself. So you have the nucleotides, right? In DNA, you have all these nucleotides. I'm just simplifying them to circles. It'll insert itself between DNA like this. And that basically stabilizes the DNA a ton. So this means that normally if you add a lot of heat, right, the DNA will kind of break and melt and denature, and it'll mess up, and that's like really bad for us, right? But this DPA kind of stabilizes the DNA, so that's very resistant to heat. Then we also have what's called SASPs, or small acid-soluble binding proteins. So these guys, they're also going to bind to the DNA. And this time they help the DNA condense down into this really compact region, which also helps protect it from radiation from the outside, right? And from chemicals that are trying to get in and damage the DNA. So you have these two kind of protective measures that really help ensure that the genome of the bacterium stays intact while it's in the endospore form. Yeah, so the process by which a vegetative cell becomes an endospore is called sporulation. So we can go through the steps real quick here, just an overview. So essentially what happens is first we gotta do binary fission, right, where we're dividing into two cells, and then we'll replicate the DNA. And this is unique because we'll do an asymmetric division, as you can see here, where one of the cells is a lot smaller than the other cell here. And this is where the smaller cell we call a forespore. So right now they're essentially equal. They're just like really different in size. what happens after they separate is that this spore, the mother cell, like the original cell here, right, the big cell, it literally just engulfs the spore, and it just takes it inside of its cytoplasm. So you can see that here. Now the spore has entered the mother cell, and what's going to happen is that the spore is slowly going to develop all these structures. So it's going to start forming that protein coat. It's going to start getting all that DPA, the dipicolinic acid. It's going to start condensing its DNA, et cetera, et cetera. So you can see that here. And as this spore is kind of forming the endospore structure, what happens is the mother cell slowly starts to undergo degradation, and eventually it'll just die. It'll lyse, right? It'll die and then kind of melt away, which will leave this endospore on its own here. And you now have that really protective form that even if you try to kill it with a bunch of heat, UV, radiation, etc., it can stay there. And then what happens is that once the bacterium is back into like normal conditions, it can kind of sense its environment, then it'll germinate. It's called germination, where it goes back into its original form, or the vegetative cell state. And then I don't know if you'll have to know this for the exams, but last year I think I just put this on my cheat sheet. It's just like endospores, like they can form in various regions of the mother cell. So there's a lot of different names for where exactly and what shape they create when they form here. So you have the endospore and then the mother cell on the outside, right? Yeah, so endospores, like I said, they're a significant obstacle to decontamination because they're highly resistant to a lot of harsh environmental factors. So we need to use some special methods for these that we'll get into. And then one place where endospores are particularly a problem is the food industry, especially canned food, because endospores often, like when they get into food, even after you sterilize the food, they're kind of, or you try to sterilize the food or disinfect it, the endospores are still in there, and they can actually kind of germinate again and then start making the food rot and spoil early. And they can also cause diseases such as this guy here, Clostridium botulinum. So this is a bacterium that causes what's called, if you guys have heard of botulism, right? This is a pretty common disease for like babies, but you can also get it from canned food. So some of these bacteria, the endospores, produce toxins that can be very harmful when ingested. All right, so to recap with the terminology, remember, before we go into the methods, so sterilization, killing everything. Disinfection is mostly killing everything except for endospores. Then decontamination just includes all these methods here. All right, so let's we can use heat as a way of decontamination. And here, the method we use is called autoclaving. And this is a sterilization method, so it's going to kill everything on these objects. So it's a common method that's used in hospitals and labs. So essentially, after you do a bunch of experiments, let's say you're in a lab, you do a bunch of experiments, you have like these tubes, dirty tubes, all this like kind of biohazard stuff, right? What you do is you'll place it in the biohazard bag, and then you'll put it in what's called an autoclave, this machine here. And this autoclave essentially shoots really, really hot steam at high pressures onto these objects, and it'll kill all the microbes on these objects. So that means that once you do this, you can kind of reuse those tubes for another purpose later on. So some temperatures, just to give you a sense of how these work, they range from like 121 Celsius, so 250 Fahrenheit, to 270 Fahrenheit. So we can also use hot air ovens. These are an alternative method that uses dry heat. So this time we're not using steam, right? We don't need water for this. We're just using really, really hot air, and we're circulating the air within the what do you call this? Like the internal components here, and then all that air will again kill all the microbes. And again, just to give you a sense of how hot it is, it's 320 Fahrenheit to 374 Fahrenheit. Okay, next, radiation. So we can also use radiation to disrupt the DNA of these microbes. And the specific method is called ultraviolet germicidal irritation, or UVGI. And this is where you're using UV rays. So you can see that looks pretty cool on the right here. Yeah, so UV radiation, the characteristic of UV is that it'll attack the DNA of any organism. And essentially what it does is in the DNA, it causes what's called thymine dimers. So it kind of makes the DNA bind to itself like this in between. And then that kind of makes it so that this DNA can't be transcribed, and it kind of like gets really messed up. So any organism that is exposed to a lot of UV will just die off. And the specific wavelength of light, of UV light, called UVC light, is specifically used because UV, there's UVA, B, and C, and then UVC is kind of the most intense out of those. Oh, and this is sorry. I think this is sterilization. Yes, I believe this is sterilization, but I can check later. Yeah, so you can also, instead of UV rays, you can also use gamma rays, X-rays, cathode rays. These are essentially just more intense versions of light. So you can see, if you look at this, you've probably seen this diagram in like science class, right? So you have the spectrum of light. So you have visible, you have UV here, and then you have X-ray, gamma, and then cathode rays are kind of within this range as well. They just means like electrons themselves. So these can also be used to sterilize. And all these are kind of called ionizing radiation. And essentially, to go into the physics a little bit, like the wavelength, right? So if UV is like this, then ionizing radiation is even shorter like this, and it basically has a higher energy that is more penetrating and more damaging than UV. Yeah, so these guys will sterilize and kill everything as well. And they can be used in food production, but it's kind of limited because it's a costly method, and it's not really suitable for widespread use. So like you can see this like big kind of, and you also like you need a lot of like protection around it. You can see these thick concrete walls, right? Because you want that radiation to get out and affect the humans that are there as well. So yeah. Okay, filtration, here's one. Again, this is sterilization, but this time we're not actually killing the microbes, we're just separating them out using a filter. So we can use these for like pharmaceuticals, like drugs that are in liquid form that are sensitive to heat. So we don't want to just like stick them in like an autoclave or something, or some method like that, because first it would just evaporate away probably, and then also like you don't want to mess with the chemical composition of that drug either. So you're separating them out with a filter. So you get these like special specifically designed polymer filters that have really, really tiny gaps in between them that are around 0.2 microns. So 0.2 micrometers. Right, so this is like 0.2 meters times 10 to the negative 6 here. So you can imagine like this isn't the most reliable method because some of the pores, like first of all, this filter itself might be a little uneven and let some of the microbes through. Then sometimes you might get really small microbes that like happen to slip through somehow. So this isn't really the best method. But yeah, and 0.2 microns, just to give you a sense of scale, right? Our cells are probably, first bacteria are probably around 1, for the most part, 1 to 10 micrometers or 1 to 10 microns. Then our cells is somewhere around 50-ish microns, or 50 to 100 for human cells. Okay, and then the kind of most straightforward method of decontamination. First, we're going to look at disinfectants. So these guys don't kill everything. Endospores can still survive for the most part. So we have 70% ethanol / isopropanol, just 70% alcohol, essentially. So these guys, they'll mess with the cell membrane of most organisms, and they also denature the proteins, so they make the proteins all different shapes and like messed up, and so they can't function properly. Okay, next we have chlorine compounds like bleach, chloramine, right? It's like the chlorine you put in your pool, for example. These guys, they're going to react. So essentially chlorine is like a very reactive molecule in general, so it kind of goes and it'll just like bond to a bunch of things in the cell, and it'll just mess them up a lot. And that's kind of the common theme for these last few. So again, formaldehyde is the same thing. It's very highly reactive, goes into your cell, or goes into a bacterial cell, start bonding, start reacting, start oxidizing things like randomly, and that just like messes with all the functions here. Then phenol or phenol, either way you can say it, it'll specifically disrupt the membrane or cell wall, so those surface structures. And you can also use it as an antiseptic, right? So you can apply this. Sometimes it's used on the skin to like reduce itching or like prevent infection on your skin. Okay, so now we're looking at sterilizers. So remember, killing 100% of the organisms. So we have, a bunch of different names that, you can just write down on your cheat sheet or something. But performic acid and peracetic acid, these are both cell wall disruptors. But sometimes this performic acid is preferred because peracetic acid, once it's broken down, it can actually produce acetate. And acetate, some microbes can actually use acetate as like a food source. So it could potentially be beneficial to the microbes. So like usually I think this is more preferred. But anyways, yeah. So then we have hydrogen peroxide. We have what's called radicals. These are essentially, if you know chemistry, they create oxygen radicals, reactive oxygen species. These are like oxygen atoms with like unpaired electrons that are highly reactive. They'll go and like kind of oxidize a bunch of things and like destroy them pretty much in the cell. And ozone is kind of the same idea. Ozone will also form these like reactive radicals, oxygen radicals. And then ethylene oxide, again, highly reactive, kind of does the same thing. Then formaldehyde, you can actually, if you put it at a higher concentration, so if you use more formaldehyde right before it was a disinfectant, but you can also make it into a sterilizer by increasing the concentration. And yeah, that's it.